Good evening, everybody, again, and welcome to this If Oxford event tonight, NeuroTales. You're in for a treat. There are 4 people ready to give fascinating stories of their roots into their scientific career, some of the motivations along the way, and some of the kind of twists and tales that hopefully you'll enjoy. It's a pleasure to pass on to Corinne Piekma from the Wellcome Centre for Integrative Neuroimaging, who's going to be looking after this NeuroTales event tonight. So Corinne is here to arrive. There we go. Thanks, Corinne. Over to you.
Thank you very much, Dane. Hello and welcome to NeuroTales. As Dane said, my name is Corinne Piekma and I am your host this evening. Tonight we explore inspiration, encounters with something or someone that awaken in us an enthusiasm to do or pursue things we've never considered before and that allow us to push beyond what we know. Our storytellers have all had inspirational encounters that set them off on their paths into science. 3 of them now work on the many facets of brain research, from understanding the networks and structures that support our behavior to the development of ever-evolving techniques that we use to study the brain. But we end the evening with a tale about a completely different but no less life-sustaining structure. Our first storyteller this evening is Hannah Willis. Hannah felt destined for a career in the arts and was always told she was arty and brilliant at English and history. But it was her history teacher who ultimately inspired her to study psychology and become interested in intricate workings of human vision. Before I hand over to Hannah, just a reminder that, as Dane said, we will have some time for the Q&A for a question after each story, and just pop your questions in the Q&A box and we will hopefully get to them. Now, without further ado, here is Hannah telling her NeuroTale.
Hello, I hope you can hear me. So thank you for the introduction, Corinne. My name is Hannah Willis and I'm a PhD student here at the Wellcome Centre for Integrative Neuroimaging. And today I want to talk to you about how I got into neuroscience. So what did I want to be when I grew up? A neuroscientist, a biologist? No, a historian. And this is going to be the story of the 3 mentors who led me here. I loved my history class at GCSE. Our teacher was fun and unconventional. He started each lesson with no PowerPoint, no notes, a clean whiteboard, which he quickly filled with stick figures of each of the different events that we'd be learning about. You can only imagine what the trenches of the Somme or the increasingly tense events of the Cold War looked like with stick figures. It's not something that you forget in a hurry. Although he was an exceptional teacher, he was also an inspirational mentor to me. At my parents' evening that year, he took me aside and he told me that he had high hopes for me and he thought I should study history at Oxbridge. This was the much needed push that I needed at the time. And so with that in mind, I was convinced I wanted to be a historian. I applied to do my A-levels. I decided to study history and English and had to pick 2 extra subjects, which I decided would be biology and psychology as an extra, a subject I'd never studied before. I quickly realized that I preferred psychology and decided to apply for university. I didn't actually listen to any of my history teacher's advice. I didn't apply to Oxbridge and I didn't apply for history. Instead, I applied to the University of York. This was mostly driven by the course, but also a little bit by the distance from home. I was desperate to get away from home and start off on my own. Psychology is a broad subject and A-level had only ever scratched the surface. I soon realized that psychology encompassed a range of topics from consciousness to the senses. It's also often misunderstood. I've often been asked whether I can read minds, explain a science fiction film, or even give advice about usually a child's mental health. York is a beautiful city. It's overrun by geese and ducks, so much so that students are often scared of entering buildings because of aggressive geese. Buses are held up by ducks crossing the road, and I was given a little toy duck at my graduation, so I'd never forget. It was here that I realized that I was actually interested in vision. This is the dominant human sense, and I was very quickly hooked on this very biological side of psychology. This was mostly driven by my second mentor, a well-known and colourful lecturer at the University of York. His lectures made sense to me, and I enjoyed the comedy and historical teachings. Who knew that by learning psychophysics, I would also be learning about Greek mythology? It was him who encouraged me to apply for PhDs and to explore my interests and vision. He encouraged me to move to different cities and take up jobs in different labs to learn about the different areas I was interested in, from colour vision to blindness. By this point, I was sure that I wanted to do a PhD, but I also wanted to travel. This is my other passion, and I also have an unusual love of snow. So I spent 1 year traveling around Asia and Europe, and I even lived in a mountain village for 5 months. This was an amazing experience. It made me more aware, respectful, and curious, but also it made me sure that I wanted to be in science. I really missed science whilst I was away, and I was so excited to come back and begin studying again. I'm now in the second year of my PhD, studying here at Oxford in a clinical neurosciences department. I'm studying blindness and after stroke, and it was here at Oxford that I met my third mentor, my supervisor, who has already encouraged me and supported me through so much. One of the main reasons that I was drawn to this department, the Wellcome Centre for Integrative Neuroimaging, was because of the female scientists, from my supervisor to even the head of our department. These are leading researchers doing groundbreaking research across fields. I'm so fortunate to have had so many incredible mentors and don't believe I would have ended up here if it wasn't for them. My colleagues are often surprised that I come from a humanities background. It's quite unusual, as many of them have always dreamed of being scientists. They can list off male and female role models that they wanted to be growing up, although these do tend to be people whose parents or even mothers were scientists themselves. I was recently asked which female scientist I wanted to be when I grew up, and I was so surprised. Science really hadn't felt like it was for me as a child, and I certainly didn't have any role models. I read an article yesterday that seemed very relevant to this talk, and apparently there isn't one woman's name in the GCSE science curriculum this year, and only half of the population could name a female scientist. It can therefore be no surprise that I thought that science was the subject for my brothers and humanities is the subject for me and my sister. Me and my sister often heard comments like, "You're only girls," when we couldn't do something as well as our brothers, despite both of our brothers being older than us. Although this was said as a joke, you can only imagine it encouraged the split between us. It's not that we were bad at science, but we were told that we were good at humanities, and so that's what we explored. In the end, we all studied sciences, my sister and I anthropology and psychology, and my brothers engineering and maths. Both of my brothers are also incredibly creative, but I believe that they were pushed towards science. My brother recently told me that he had wanted to study maths and art together at A-level. He thought those subjects went perfectly together, but he was told by our sixth form college that he wasn't allowed. A weird policy at the time. For some reason, we learn that there is a dichotomy between art and humanities, maths and science. But I believe that they're not so different. A lot of the abilities that we learn in humanities subjects are also important for science. For example, to be a good scientist, you need to be creative, you need to be a good storyteller, you need to be a critical thinker, a writer, a communicator. These are all things that we learn in humanities and art subjects as well. So to conclude, I believe that science is for everyone, because science is just the story of how things work. And if we use some of these humanities skills, like communication and critical thinking, then we will open up science to everyone. So as I've explained, I wanted to be a historian and ended up a visual neuroscientist. I believe it's never too late to change your mind, especially with a little help or encouragement.
Thank you so much, Hannah, for that very personal and emotional story. So in her research, Hannah tries to understand what happens in the brain when people lose part of their vision after a stroke or a head trauma. She hopes that this knowledge can inform the rehabilitation and therefore have sort of better outcomes for patients. So Hannah, we do have a question for you. What do you think is the best way of, or a good way of getting more girls into science?
That's a really good question. And I think just exposure, I'd say, is sort of events like this where young girls can see other incredible scientists and see what they're doing and learn about the possibilities. Because I think that one of the problems for me is that I didn't know that it was a possibility. I didn't know what neuroscience was, and I definitely didn't know that that was something that girls could do. So I think, so one of the things that we do like to do here at the Wellcome Centre, we go into schools and we run events and things like that. I think that those sort of organisations that will help to encourage young girls to sort of see other role models and kind of be inspired to hopefully choose those subjects themselves.
That's brilliant. Yeah, I think you're completely right about that. You can't be what you can't see, said someone once to me, and it just does, it's just so obvious, really. Anyway, thank you so much for that, Hannah. We will now be going on to our next storyteller, Christiana Tisca. Are you there, Christiana? Christiana. Hello. Christiana's journey into life as a physicist started actually with a love, also with a love of words and stories and novels and creative writing. And she argues that actually those are essential skills to be a good scientist. So here is Christiana telling her NeuroTale.
Thank you, Karine. Hi, everyone. My name is Christiana Tisca, and I am a PhD student doing research on brain imaging. I work with very big magnetic resonance imaging scanners, which I use to scan mouse brains, which I've learned over the past year since I started doing my PhD that they are much, much tinier than human brains. They're actually roughly the size of your index fingernail. I'm originally from Romania, a country in Southeast Europe, which is often confused with Bulgaria, and whose capital city, Bucharest, is often confused with Hungary's capital city, Budapest. When I introduce myself, I like to say I'm from Transylvania because it sounds a bit spooky, which is fit for October, right? And because it makes me sound more interesting, or at least that's what I like to think. So why am I here today? So today I want to share with you what, or more specifically who, inspired me to pursue a career in research, and what I believe are essential skills that researchers need to have. Looking back at how I ended up here, I realized that I wouldn't be here if it were not for a number of brilliant teachers I have had over the past 15 years. So let me quickly take you back 15 years ago. This is when I met my Romanian literature teacher. And I know you're probably wondering, didn't I just say I study brain imaging? What does literature have to do with brain imaging? Just bear with me. My Romanian literature teacher was a quiet, strict young woman who had just graduated from university and was extremely passionate about literature. She wanted to teach us much more than the curriculum allowed, from the world literature to relatively unknown Romanian poets. She also encouraged us to read and keep a reading journal and present our most favorite works in front of the class every term. She put together a literature club as well, where she encouraged us to write our own works, from short poems to plays or even short novels. My works were mostly love poems about meeting my crush in the hallway. But looking back at that, I realized how similar this sort of event is to our day-to-day lives as researchers, where we often face the challenge of presenting our work in front of our peers, or writing a paper about our most recent findings. Also, I think science can engage everyone if it is thought of as a story. In the words of my Romanian literature teacher, as scientists, we should ask ourselves, what is the message that you want to share with your audience? What's your plot twist? After all, I think science has a magnificent story to tell, which is the story of our universe and everything that lies within it. But please don't let me become too metaphoric, and let me continue with my story. After I finished gymnasium, which is primary school in Romania, I decided, aka was pushed by my parents and teachers, to study science in high school, even though coming from a small town school, I was only vaguely familiar with the concept of science. I also at the time really wanted to continue writing my poetry. In the weeks after starting high school, I realized that amongst all subjects that I was taking at the time, I specifically hated physics. And this was because I was probably never going to understand how it works. But I had a chance of having a great physics teacher without whom I wouldn't be here today. We stayed in touch over the years, and I even invited her to this talk. I hope she is listening right now. She opened up my appetite for the story of physics. I remember her story of about being the only woman physicist in a class of hundreds of physicists at the University of Bucharest, and about how she didn't let that put her down, topping her class by the end of her studies. This really drove me to work really hard and study physics. And with her support, slowly but surely, I started getting to grasps with the concepts of optics and mechanics and thermodynamics. And I loved the subject so much that I decided to study physics for my undergraduate studies. My chosen specialty was going to be astrophysics, but I ended up doing a PhD in brain imaging, which I agree is a bit different, but I would argue it's also quite similar in a way. I went from wanting to use very big telescopes to look at the biggest structures in our universe to now using very big magnets to look at some of the smallest structures inside our brains. Physics connects these seemingly very different areas and is essential to understand both the outer space and the smallest structures that make up living creatures. And I know you may not have thought that studying the brain requires much help from physicists, but the truth is that most areas of science today find themselves needing the knowledge of people who have trained in apparently unconnected areas. What I'm realizing more and more is that the progress of today's neuroscience needs the help of a variety of scientists. For example, we need physicists to make sure that we get the best images that we can. We need the help of biologists to interpret those images. And we need the help of computer scientists to write programs to analyze that information. A research team of people with a diverse set of skills has a great advantage of being able to approach a problem from many different perspectives and is much more likely to solve that problem in a creative way. And I myself am very lucky to be part of such a team at the University of Oxford. Finally, regarding my future, I really don't know at this stage if I want to continue being a researcher for my whole career or even if I'm capable of that. But I know what I need in order to continue down this path. And that is seeing and working with people like me. Unfortunately, gender imbalance and recognition in academia are still issues that we need to work on. I, for one, have learned that I have a place here by working and interacting with women researchers, including my supervisors, who have inspired me deeply. I'm also currently trying to do my part to help with this by helping coordinate a group of women in physics and engineering within my center. Within the group, we meet every couple of weeks and have an invited speaker usually. And we organize meetings where we discuss issues which are very important for women working in research and academia, such as job opportunities, caring responsibilities, or job applications. Here, we aim to learn from each other and openly discuss the problems that we face. And I think that this kind of group is really important for people belonging to all kinds of minorities, as it offers a safe place for people to meet and get to know people like themselves who work in academia. I think this is essential for the progress of science. I believe that we learn most when we relate to our teachers and mentors. Science is a place where everyone should feel welcome, a place where you can find someone like yourself. I'm very lucky to work with brilliant women I can relate to, but I am aware of the work that we still need to do to make sure that any budding scientist can identify someone like themselves working in research or academia and can tell themselves, well, if that person can make it, so can I. Thank you.
Thank you very much for that beautifully told story, Christiana. Christiana tries to understand the link between our genes and their influence on brain structure in humans and in other animals. Christiana, we do have a question for you. So you look for something called biomarkers in the brain to understand the structure, but what are biomarkers and what do they tell us?
I think, well, biomarkers can be anything that is related to the data that you acquire, and it's something that is characteristic to that particular data that can help you get more insight into that information. So for instance, you know that a particular measurement that you're making is much bigger in people who are suffering from a particular disease. So that particular measurement can be a biomarker of disease.
So does that mean that you can, if you see that in someone else, that could be a hint that someone else might be potentially getting a similar disease or having a similar structure or whatever the biomarker is for? I see, okay. Let's see, there is one other question. I'm just trying to work out how to ask this question. Oh yeah. So how do you think STEM and art can connect better?
This is a great question, and I think it can be answered in many different ways. I think they can both connect better if you bring together scientists and artists. But they can also come together by asking scientists to create art as well. I think anyone can do art. And I think artists can also engage with science as well. I think this is something that we're trying to do now as well, aren't we? Just we're trying to share our, as scientists, our knowledge and impressions about science with people from a wide variety of background. And hopefully this can inspire artists as well.
Okay, so thank you very much for that. We will now have to move on to the next storyteller, and that is Beatrice. Beatrice had an overwhelming sense of wanting to give back to the country where she grew up in, and that is inspiring her to do everything she does. And so here she is talking to you about telling you her NeuroTale.
Hi everyone, my name is Beatrice, and I'm a PhD student in clinical neurosciences studying non-invasive treatments for tremor. So I'm about to tell you the story of how I ended up here in Oxford. It all began when an armadillo skeleton was found in the woods near my school. My biology teacher called me to the lab and told me, I have a surprise for you. He gave me my first lab project, assembling the armadillo skeleton. I was in 5th grade and I had just started volunteering in the school lab. I was born and raised in Jundiaí, in the countryside of São Paulo, Brazil. My mom is a geography teacher and I grew up seeing her passion for teaching and for supporting every student. Her work gave me a scholarship at one of the best schools in town, and she always told me to take every opportunity I had there. With that in mind, I joined the lab group as soon as I could. There, I started to learn how to think as a scientist. I was 11 years old when I began dreaming about becoming a scientist and contributing to the development of Brazil through science and technology. In 9th grade, I got a scholarship to a big school in another town, and there I found out that it is possible to study in the best universities in the United States. In the US, I would be able to explore all my passions from science to music and history with the liberal education system of the US, while also doing scientific research. But what mattered the most for me was that I would have even more resources to have a shot at impacting Brazil through science. So at the time, my English was terrible. I didn't know much beyond the to be verb, but I was very motivated to learn so that I could apply to college in the US. One secret is that you don't really need to be fluent in English to do well in standardized tests to US universities. You just need to be really good at taking exams. So after dozens of practice tests and memorizing all the patterns in the questions, I was able to get the score that I needed, and I was admitted at Brown University. Being there, however, was a totally different story. I could, of course, read, write, and understand English, but speaking was just so difficult. During my first year there, I felt that I wasn't able to be myself in English. Also, I had never taken any academic classes in English before, and I felt that I was learning a new language in addition to the course materials. Added to that, dealing with cultural shocks and the cold, that was my first experience with real winter and temperatures below 10 degrees Celsius. My first year was challenging. I remember walking on the snow without a proper winter coat, without proper shoes, and it was so cold that it would hurt to breathe, and my nose was bleeding. However, that was also the year that I took my first neuroscience class. I initially struggled a lot with the new vocabulary in English, but I quickly became very passionate about it, particularly when it came to learning about motor control, or how thought is translated into actual movement. I was scared of studying neuroscience though. In Brazil, it was not even an undergraduate course. So how do I impact the country with a field that was so new? I knew of one big reference in the field, Miguel Nicolelis, a Brazilian neuroscientist who is also a professor in the US. He was a leading reference in a field called neuroengineering, which involves using engineering tools to better understand the brain and to bring back brain functions that were lost due to injuries or diseases. I found out he was going to speak at a conference in Boston, near where I lived, and I decided to go there just to ask him for some career advice. He gave the most inspiring talk. He presented one of his neuroengineering initiatives, the Walk Again project. This involves an exoskeleton controlled by brain signals collected from electrodes in the surface of the head. It was developed for paraplegic patients who had no movement or sensations on their legs due to spinal cord injuries. The patients would think about moving their left or right legs while wearing this metal structure called exoskeleton, and then the exoskeleton would move their left or right legs. He also described how he built from scratch research centers and schools in one of the poorest towns in Brazil, Macaíba, and boosted the development of the area. He developed the projects with a model of a scientific education for low-income children, many of them the first in their families to ever go to school. At the time, Brazilian science was starting to go through a rough path with regards to cuts in funding, and it was just so inspiring to hear about his work. When I looked behind in the audience, other Brazilians were literally crying. Nicolelis' work was a proof of concept that we can transform Brazil through science. I was hoping Nicolelis would stay for the coffee break so that I could have a chat with him, but after the talk, he just got his things and left. I remember sitting there thinking just, oh no, like I went there just to talk to him. So I got up and I ran after him. I introduced myself, and then the most amazing thing happened. He said, by the way, I have some research opportunities, some internship opportunities in my laboratories. That was insane. Nicolelis was a hero for me, one of the main references among Brazilian scientists, and someone I would see in the news when I was a little girl dreaming about becoming a scientist. So in the summer after my first year of undergrad, I worked in the Walk Again project laboratory in São Paulo, where the exoskeleton for paraplegic patients was being developed and tested. That was an amazing experience. Something that had never been seen before in the history of medicine happened there. So after training under the protocol for using the exoskeleton, every single patient recovered at least some movement or sensation of their legs. It was so inspiring to see that happening, to see people who were paraplegic being able to move their legs again. It also brought me a lot of faith in Brazilian science and showed me how we can impact the country with neuroscience. I finished the internship determined to work in neuroengineering, and I planned the rest of my undergraduate education, all of my classes, to develop skills for this research field. I ended up getting a double major in biomedical engineering and neuroscience. Doing a PhD had always been part of my plan since I was in high school, so it was natural that I decided to apply for PhD programs in my last year of undergrad. However, many laboratories in the US in my field are focused on developing the most advanced possible technology in the field, which results in cool science projects, but it costs millions of dollars per patient, so very few people can have access to that. We can say that science is universal and that regardless of where I work, I could impact Brazil, but I don't really think that's true for medical technology. I knew that high cost neurotechnology is very unlikely to reach developing countries like Brazil. It wouldn't even be an option for most people from low-income backgrounds in developed countries. And then I found a laboratory here in Oxford that is concerned with making neurotechnology more accessible. So I decided to apply here. My PhD project focused on developing non-invasive technology that means without the need for surgery or medication, or something that you can just wear on the surface of the body to reduce tremor or shaking in conditions like Parkinson's disease. Today, treatment options for tremor involve inserting an electrode into the brain, which can work very well, but of course, most patients cannot have access to that since it involves neurosurgery and that is expensive. Another option is medication, which can work, but sometimes leads to side effects or helps with some symptoms of conditions like Parkinson's, but not with the shaking itself. So there is a clinical need for non-invasive treatment for tremor without surgery or medication. I'm trying to develop something like a wristband or a bracelet that patients could wear at home or in the streets, and that would reduce their tremor. This is a project that I'm really excited about, as I see potential for it being affordable and accessible to people from lower income backgrounds, and that is something that could be used in developing countries like Brazil.
Thank you so much for that, Beatrice. So Beatrice currently collects data that helps her to understand her patient's tremor. It also stimulates the nerves with on the outside with tiny electric currents that she applies to the skin of a patient's arm to look at how she can sort of change the tremors with that stimulation. Beatrice, we do have a question for you. How long do you think it will be before the bracelet it would be available to patients with Parkinson's disease? How far are you into your research?
Well, that's a great question. And I just got started this past year. And from January, I will start working on developing the actual bracelet. So far, the whole setup takes up like half of a room, like the size of a couch approximately, and you definitely cannot wear that in your arm. And I'm working on developing the actual wristband from January. And I would say it would probably be a couple of years until that would be widely available.
Have you got any sense of how you're going to make it smaller?
Yeah, yes. It is a big engineering project, but involves like putting all the sensors within like a small contained space and summarizing all the circuits in a small structure. So yeah, and I'll be working on that from January. And finish the first steps of the tests with the bigger setup.
And it would have an incredible impact on Parkinson's disease patients all over the world, I suppose. Like,
yeah, so that's what I'm really excited about, because this is something that would be accessible, would be easy to wear, lower costs than other neurotechnology currently available. And so, yeah, I'm very excited about it and very hopeful this will be helpful and applicable for many people.
Super. Thank you so much for telling your story to us. I just wanted to go back to one question from Susanna, which was actually for Hannah. And it is a question to do with stroke, Hannah. I'll wait until you're here. Could there be a way to fix your sight after stroke?
Well, that's a very good question and is basically the focus of my PhD. So, but firstly, I think just to tease apart the question, Fix your sight is quite a difficult thing to do. So it's not a word that I would use. I would probably say that we try to improve vision. And that is something that I do believe is possible. I'm not sure whether that's the same as fixing your sight, but there has been a lot of research done in the States that's been shown to improve vision. And we're actually collaborating with a team in Rochester in the United States who have shown a lot of that research. And we're sort of bringing it to the UK to run trials here, but with neuroimaging. So we're particularly interested in the neural correlates of any improvements. So quick answer, yes, potentially.
Okay, thank you very much for that, Hannah. In the meantime, we've also just had another question for Beatrice. Beatrice, would you be available to answer that question still?
Yeah,
so the question is, how would the tech in the bracelet work, and does the type or stage of Parkinson's disease affect the device's ability to work?
So for the first part of the question, those are both great questions. So essentially, we will record the tremor in real time, like it would be continuously recording the tremor, and measure some characteristics of the tremor that are intrinsic to each patient, and they actually change over time a lot in Parkinson's. So even like for the same patient, the tremor changes within seconds. So sometimes the tremor is like this, then suddenly changes to this, then suddenly changes to this. So we need to have a device that can detect how the tremor is behaving, which are these characteristics, and give the specific parameters for the stimulation, the specific characteristics for the electrical stimulation, according to the patient's tremor. So it has to be something adaptable, which is actually like one of the big challenges with that. There are like other options of like non-invasive technology being tested by other groups at the moment, but none of them is adaptable on the sense. And there is a lot of variability in Parkinson's tremor, so we need to account for that in the device. And so with regards to how the stage of the condition would affect how the device works, this is something that we're still to find out. And so the first stage of my project, as I mentioned, showed us that there is a lot of variability in the tremor. It changes a lot from patient to patient, and even for the same patient within seconds. And we need to look more into that. And for that, we need to be able to record the tremor for longer periods of time. And that's what I'm going to do. Like I am, from January, I'll be developing the actual device the patients can take home, so we can record the tremor for extended periods of time. You can think of tremor as a wave, like an oscillation. And so tremor has like a frequency, it has an amplitude, just like a sound wave, for example. And the device needs to be able to measure that and give characteristics for another wave, which would be the electrical stimulation, according to each patient's tremor. So we don't know how the stage of the disease would affect the effectivity of the device, but we want to make something that will be adaptable to every patient.
All right, fantastic. Thank you very much, Beatrice. So we will now move on to our final storyteller of the evening, which is Andrew Galloway. Andrew is a botanist and a horticulturist and loves everything to do with plants. Tonight, we end with his meditative story about the brain of the forest, in which he explores the network that connects all the trees on Earth. Andrew?
Imagine walking through a forest, a psychedelic kaleidoscope of leaves that have started their endless march into autumn, turning into a golden brown colour which surrounds you. You close your eyes as you walk on the hardened, uneven ground. You feel the gentle autumnal breeze on your glistening cheeks. You begin to focus your attention on the rustling of leaves that have fallen from the surrounding trees. No other sound can be heard in the forest except the cool breeze. You stop and inhale a breath of fresh, untainted air. You then gradually open your eyes whilst gently exhaling, taking in the comforting peace that surrounds you. For a moment, worries of debt, job security, and illness fade. Your mind seems to have cleared out all of its toxic, negative thoughts. None of the mundane stresses of everyday life disturbs you, whilst you absorb your surroundings. Continuing on your journey, you notice a small clearing on the forest floor. You slowly bend down to investigate this curious clearing by brushing away the crisp brown leaves. You start to wonder what lies beneath the loamy soil. You push away more of the topsoil to further cure your insatiable curiosity. As you delve deeper, you start to see a crisscross network of wiry roots that almost resembles a bustling jumble of highways. As you continue to scrutinize this network, you begin to appreciate the complexity in this tantalizing matrix of root and soil, which now seems to resemble a telephone exchange. On further scrutiny, you see an additional sub-network of white tentacle-like hyphae that spans the roots. You start to wonder what the purpose of this network is and how far it stretches. You continue your investigation in the serene forest. As you look around, you spot a small whip-like maple tree that waxes and wanes in the fresh autumnal breeze. You slowly walk towards this trembling treeling. You crouch down to clear the heaped crisp leaves at its base. As you clear the leaves and the soil beneath, you start to see the matrix again, but it contains larger, thicker interconnections of hyphae. Out of curiosity, you decide to follow this network containing the larger interconnections to what you hope leads to an emerald surprise. This emerald surprise turns out to be a grand oak matriarchy that looms over the surrounding area. To your amazement, you realize that this network links the smaller, weaker maple with the grander oak. You start to wonder how this treeling survives when its neighbouring larger trees gobble up the surrounding light. For once in a long time, you're feeling that your mind has something to contemplate other things than the constant bubbling of anxiety and melancholy. Your renewed enthusiasm spurs you on to learn more about this forest network. In fact, the grand old oak feeds the younger maple independent of species. These trees form one minor component within a large breathing organism that is the forest. This wood wide web not only shuttles food, but also exchanges information similar to that of the World Wide Web, particularly warning of impending dangers. The WWW blankets trees from harsh weather and a lack of resources needed for their growth. This vast ancient network connects the vast majority of plants within the forest through a primordial marriage of fungi and plant, which is the longest lasting marriage predating all life on land. All these connections within the biosphere of Foul Earth form a greater organism that we have managed to divorce ourselves from within our gleaming modern cities in sterile dwellings. Some know this greater life form as Mulagina, which is the only oasis in the obsidian void of the cosmos. From the smallest bacteria to the largest seafaring mammal, we're all strands within this network that is Gaia. We can easily forget or ignore within our modern day distractions and stresses. Neglecting Gaia can cause us to feel anxious, similar to what a lost child might feel. And only by rejoining her, we can preserve our well-being and prized oasis. And that's the story.
Sorry, my camera was a bit slow. Thank you so much, Andrew. I can't help but think that we could do better in supporting the weakest in our society, like the trees do. That would be a good thing. Andrew is currently a research coordinator at our centre. And Andrew, we also have a question for you. So how was the I can't actually say it. How was the Wood Wide Web?
The Wood Wide Web, yeah.
The Wood Wide Web actually discovered?
I think it's one of those serendipity sort of situations where someone was exploring soil and the matrix and what sort of life was involved using sort of sequencing methods. And they came across a fungi species, which was very dominant in that sample. And then they carried on sampling across the forest and realized that the actual sample was the same thing. And they delved deeper and they took root samples and found that this sort of Hyphae Matrix was actually growing within the roots and was actually in the trees as well, and other plants. But unfortunately, when the scientists were looking into this, they actually destroyed most of the matrix. But there's new novel methods of imaging sort of in situ. You don't have to destroy it. But that's sort of, I think that research was done in the north of the US, that sort of research in Oregon.
And so you say, like, are all the trees connected all over the world?
Most trees are connected within the forest, and it's sort of like a society almost. If one tree needs resources, this fungi, and we don't know how it controls it, actually takes resources from a larger oak to a weaker maple, and it could be reversed as well. It can also shuttle water to parts of the forest that are very dry. Some plants can actually feed off the system and take too much. Some fungi can actually stop this. So it's really fascinating, but not much is known about this. And in fact, in the plant world, we know more about the moon than Mars compared to how plants grow and form this network. It's fascinating.
I guess that is true. So we have another question for you. How do you think climate change would affect the network, or do we know how it affects the network?
It's a really interesting question. There's a lot of modelling, but unfortunately it neglects this sort of network. So it's very underdeveloped, but there's a sort of hypotheses out there that plants will then start rejecting. So in farmlands, let's go to farmlands where you give a lot of nutrients, plants actually reject the network because there's already enough nutrients. And that actually helps to destroy the soil eventually through soil erosion. So not many roots falling into the soil, and it blows off. And we think with climate change that there's going to be a speeding up of the nutrient cycle. So then plants don't really need so much fungi, so they reject the network. But there's also a lot of theories that they actually need the network more and overload it and it crashes. So it's still hotly debated, but we can surmise that it's not going to be a good effect.
Okay. And we have one more question, which I will sort of distill down. It's a question from Sienna, who's asking that she thought that, or she or he thought that the trees would only help members of their own family, but as you said, that doesn't turn out to be the case. So why is it beneficial to trees, to help trees from another species? Do you know?
That is also a very, very good question. And the simplest questions, as we all know in science, are the hardest. It could be some sort of soil stabilization effect. Generally, trees of the same species don't want to be next to each other because then they have to compete and it's a nightmare. It could be a secondary effect. So the trees are actually trying to feed their own species and the fungi because the fungi controls this network, actually just takes resources, gives it to the tree, and if the tree grows bigger, then it needs more of a network. So it's almost like a shopping sort of capitalism system there. But again, we don't really know. We know we have a network. We know it's independent of species. We know that the fungi controls it, but that's about it. So it's a very interesting question.
Okay. Well, thank you so much for that very beautiful story, Andrew. And thank you to all our storytellers this evening. We really hope you enjoyed our stories, and we hope to see you again sometime for more NeuroTales in the not too distant future. Thank you very much for listening. And now back to Dane.
Thank you, Corinne. That was a really fascinating group of people and some really good questions. Thanks again for coming, and thanks to all of our speakers. Have a good evening, everyone. Bye-bye.